Now, why is this glycine metabolism important? From your exam point of view, a couple of questions have been asked in exam. These include enumerate compounds derived from glycine, describe creatine biosynthesis from glycine, explain the glycine cleavage system, what is Lohmann's reaction, and what is its clinical significance. From patient point of view, there are two inborn errors of glycine metabolisms, and these include non-ketotic hyperglycemia and primary hyperoxaluria. Now let us try to figure out answers for all these questions in this. Now glycine is a non-essential amino acid. It is synthesized from serine. Now serine loses its methylene group to tetrahydrofolate, which gets converted to methylene tetrahydrofolate, and serine gets converted to glycine, and the enzyme is hydroxymethyl transferase. Now glycine is glucogenic amino acid. Now glycine accepts the methylene group from tetrahydrofolic acid and gets converted to serine. Now here tetrahydrofolic acid is one carbon compound and it is a carrier of all the different one carbon compound. Here with respect to glycine metabolism, methylene is the group that is carried by tetrahydrofolic acid. So glycine accepts the methylene group from tetrahydrofolic acid and gets converted to serine. Now this serine gets converted to pyruvate, and pyruvate gets converted to glucose by gluconeogenesis pathway, where glucose is synthesized from non-carbohydrate source. Now the pathway is as follows. Pyruvate gets undergoes carboxylation from oxaloacetate. Now this undergoes decarboxylation to form phosphoenol pyruvate. This gets converted to phosphoglycerate. Now these are all reversal of glycolysis. This gets converted to 3-phosphoglycerate and phosphorylation to form 1,3-bisphosphoglycerate. This gets converted to glyceraldehyde 3-phosphate, which gets converted to fructose 1,6-bisphosphate, and this gets converted to fructose 6-phosphate, followed by glucose 6-phosphate and glucose. So glycine, because it forms glucose, it is considered to be a glucogenic. Coming to other functions of glycine, and the question can also answer to this question, that is compounds derived from glycine. These include heme. Heme is derived from glycine. Now glycine condenses with succinyl-CoA to form delta amino-levulinic acid. 4 molecules of delta amino-levulinic acid get converted to phosphobillinogen, which gets converted to urophosphorinogen, coprophosphorinogen, and finally to heme. Heme combines with globin to form hemoglobin. Now glycine is also involved in the biosynthesis of tripeptide glutathione. So glycine condenses with cysteine and glutamate to form glutathione. Now this glutathione is a tripeptide. This is required for maintaining the RBC membrane integrity, and it is also required for the transportation of neutral amino acids, which include phenylalanine, isoleucine, leucine, valine, tyrosine, tryptophan. Now through a special pathway called Meester cycle. So these are the functions of glutathione, which can be asked as a 3-marker question, or it can be asked as a 2-marker question. Now glycine is also an inhibitor neurotransmitter in brain and spinal cord. So this will result in membrane hyperpolarization by opening the chloride channel, and this is how it decreases the neuronal excitability. So apart from this, glycine is required for conjugation of bile acids. Now these bile acids are derived from cholesterol. Cholesterol is a 27 carbon compound, and this gets converted to cholestane, 24 carbon compound, and this undergoes hydroxylation to form cholestane diol, wherein 2 hydroxyl groups are attached, and cholestane triol, wherein 3 hydroxyl groups are attached. Now cholestane triol gets converted to cholic acid, and cholestane diol gets converted to quinodeoxycholic acid, and these two are known as primary bile acids. So you can use a mnemonic to remember this primary bile acid, PCC. P stands for primary, C stands for cholic acid, the other C stands for quinodeoxycholic acid. Now these primary bile acids are conjugated by glycine to form bile salts. Now why do we need bile salts? These bile salts increase the surface area of absorption of lipids and also fat soluble vitamins. Apart from that, glycine is also required for de novo synthesis of purine nucleotides, adenine and guanine monophosphate, diphosphate and triphosphate. Now, so the glycine contributes to C4, C5 and N7 of purine nucleotides. So these are the compounds that are derived from glycine. One that is derived from glycine is creatine. So this itself can be asked as a 5-marker question for you. Now the pathway of creatine biosynthesis is as follows. Glycine combines with arginine. The reaction is catalyzed by amidotransferase enzyme, which transfers guanidino group of arginine to glycine to form guanidoacetic acid. This part of the reaction is taking place in renal cells. Now the guanidoacetic acid is transported through circulation to liver, where it gets methylated, and the methyl group donor is S-adenosyl methionine, which is active form of methionine essential amino acid. After donating the methyl group to guanidoacetic acid, S-adenosyl methionine gets converted to S-adenosyl homocysteine, and the enzyme is methyl transferase, and guanidoacetic acid gets converted to creatine. Now this creatine is released into circulation. It reaches the skeletal muscle, cardiac muscle, and the smooth muscle, where it is phosphorylated by creatine kinase enzyme to form creatine phosphate. Now this creatine kinase is an isoenzyme, and it has got 3 isoforms or isoenzymes. Now this includes CKMB, which is present in cardiac muscle, CKBB, which is present in brain, and CKMM, which is present in skeletal muscle. And this requires energy from ATP hydrolysis, and the phosphate group is incorporated to from creatine phosphate. Now, so this CK creatine kinase, the 3 isoenzyme has got great clinical significance. CKMB, which is present in myocardial tissue, it is elevated in myocardial infarction. CKMM, which is present in skeletal muscle, it is elevated in muscle injuries and various myopathies. CKBB, which is present in brain, it is elevated in case of head injuries, brain tumor, and stroke. Now the creatine phosphate that is stored in muscle, it is utilized for muscle contraction. The energy is utilized wherein creatine phosphate undergoes hydrolysis to form creatine. Again the reaction is catalyzed by creatine kinase, wherein ADP gets converted to ADP. So the initial part of the muscle contraction energy is provided by breakdown of creatine phosphate to creatine, and this is known as Lohrmann's reaction. Now clinical significance of or metabolic significance of Lohrmann's reaction is that this is a rapid ATP energy regeneration reaction in muscles, and this plays a very important role wherein it contributes to energy for the muscle contraction within first few seconds, even before the operation of glycolysis and TCA cycle. Now, so this creatine phosphate undergoes spontaneous degradation to form creatine. Creatine is nothing but anhydride form of creatine phosphate. Now, this creatine is released into general circulation, and the concentration of creatine in males is around 0.7 to 1.4 milligram per deciliter. In females, it is around 0.6 to 1.2 milligram per deciliter. Now, there is a difference in creatine concentration in males and females. This is because the muscle mass differs among men and women. Now, this creatine gets excreted by the kidney in urine, and the daily excretion of creatine is around 1 to 2 gram per day. Now this creatine is an indicator of renal function test because creatine is freely filtered by the glomerulus into the proximal tubula into the lumen, and this is not reabsorbed, and very small amount of creatine is secreted by the renal tubule cells. Hence creatine is considered to be a better indicator of renal function when compared to urea, because urea 40% of the urea is reabsorbed and it will overestimate G. Coming to breakdown of glycine, there are two mechanisms by which glycine is broken down. One, the major mechanism is the glycine cleavage system. Now glycine is the simplest amino acid with this molecular formula. It has got amino group, carboxyl group, and one extra hydrogen atom. So the molecular formula is as follows: NH2, COH, and CH2. Now this NH2 is released as ammonia by deamination. The carboxyl group is released as carbon dioxide by decarboxylase enzyme. The hydrogen atom is accepted by NAD, which gets reduced to NADH and H+, and this CH2 is taken up by tetrahydrofolic acid, which becomes methylene tetrahydrofolic acid. Now whenever this glycine cleavage system is defective, it results in a rare inborn error of metabolism called non-ketotic hyperglycemia, characterized by increased glycine in blood, CSF, and urine. So the other mechanism of breakdown of glycine mainly takes place in peroxisomes. wherein glycine undergoes transamination to form glyoxylic acid, which undergoes reduction to form glycolate, and this undergoes oxidation to form oxalic acid. So one more inborn error of metabolism associated with glycine is protein targeting defect. So if these enzymes, if they are present in mitochondria instead of peroxisome, it results in a protein targeting defect called primary hyperoxaluria, characterized by increased oxalates manifesting as renal stones, colic, and hematuria. Coming to in detail about non-ketotic hyperglycinemia, also called as glycine encephalopathy. So as I already stated, it's a rare autosomal recessive disorder caused by defective glycine cleavage system, and the most commonly affected enzyme is P protein or glycine decarboxylase. Now, so there occurs accumulation of glycine in the blood. Now this glycine has got dual action. Now it acts as an inhibitory neurotransmitter in the spinal cord, whereas in the brain it acts as agonist or excitatory effect on N-methyl D-aspartate receptors. Now when increased accumulation of glycine overstimulates this N-methyl D-aspartate receptors, it results in severe neurotoxicity manifesting as seizures, and the child can lapse into coma. There are two types of non-ketotic hyperglycemia. One is the classical neonatal form, which presents within 22 hours to 22 hours or birth, or 72 hours after birth. And what are the characteristic feature of this classical neonatal non-ketotic hyperglycemia? So it is characterized by poor feeding, lethargy, hypotonia, apnea. This is because glycine acts as inhibitory neurotransmitter in spinal cord, so there will be lethargy, hypotonia, apnea, and child can lapse into coma. Intractable seizures because of overstimulation of NMDA receptors in brain. The late onset form, the child presents with developmental delay and refractory seizures. Now, what are the lab findings in non-ketotic hyperglycemia? Obviously, the glycine is increased in blood, CSF, and urine. So, if you can remember this plasma to glycine, CSF to plasma glycine ratio, it will be more than 0.08 in this disorder. At the same time, the blood glucose is normal, ammonia is normal, there is no metabolic acidosis and no ketone bodies, hence the name non-ketotic hyperglycemia. Coming to the other disorder associated with glycine metabolism is primary hyperoxaluria. So this is a protein targeting defect. The enzymes, instead of being in peroxisome, they are present in mitochondria. So what happens is, so glycine undergoes transamination to form glycolic acid, undergoes reduction to form glycolate, oxidized to oxalic acid, and this oxalic acid combines with calcium, and calcium oxalate precipitates in the renal cortex medulla tubules, forming renal stones characterized by severe renal colic. Colic is nothing but pain, and manifest also as hematuria, presence of blood in urine. So this was about primary hyperuria. So this was all about glycine metabolism. I'm just sharing you a one wonderful technique of remembering various biochemical concepts. This is known as SQ3R technique. So S stands for survey, Q stands for question, and 3R stands for read, revise, and recall. Survey is nothing but whenever you are reading the textbooks of biochemistry, it can be either Vasudevan or Satyanarayana or Lippincott. So you have to give a very rapid reading of the textbook, and this should be followed by questioning. Why is this topic important from exam point of view or from patient point of view? Obviously, I am addressing this question part because all this I have been covering in these videos. Now, this should be followed by rapid reading. If you do not have time for rapid reading, you can watch all these videos, and once you watch these videos, close the video and try to revise the concept that you have watched in the video, and this should be followed by recall, so you can mentally frame 5 to 10 keywords to remember the concept. So the other technique I'm presenting to you is self-assessment using the MCQs. Now this self-assessment will help you to evaluate how good you have understood the topic. So here, so glycine is classified as, I'll give you a couple of seconds to mentally answer this question. 3, 2, 1. So it's a non-essential amino acid synthesized from serine. Glycine is synthesized from which amino acid? I already given the answer, it is serine. Conversion of serine to glycine requires 3, 2, 1, and the answer is tetrahydrofolic acid, because tetrahydrofolate is the carrier of one carbon compound, and serine donates the methylene tetrahydrofolate group, so it is carried by tetrahydrofolic acid. Glycine cleavage system is present mainly in 3, 2, 1. Answer is mitochondria. Whereas the enzymes that degrade the glycine are present in peroxisome. Glycine cleavage system as such is present in mitochondria. Defect in glycine cleavage system leads to 3, 2, 1. Answer is non-ketotic hyperglycemia. Non-ketotic because there is no metabolic acidosis, no ketone bodies, no glucose in blood, and no ammonia in blood. Glycine contributes to carbon atoms for the synthesis of 3, 2, 1. All of the above. Glycine combines with succinyl-CoA to form delta-amino-levelic acid and further gets converted to phosphorylinogen, urophosphorinogen, coprophosphorinogen, heme. Contributes to C4, C5, and 7 of purines, and it is required for the synthesis of creatine. Glycine is a precursor of which inhibitory neurotransmitter? 3, 2, 1, and the answer is glycine itself, because it's a inhibitor neurotransmitter which acts by opening the chloride channel, bringing about membrane hyperpolarization. 1 carbon units from glycine metabolism are carried by 3, 2, 1, and the answer is tetrahydrofolic. Glycine is glucogenic because it forms 3, 2, 1, pyruvate, and pyruvate forms glucose by gluconeogenetic pathway. Which of the following requires glycine for synthesis? 3, 2, 1. Porphyrins, yes, because heme synthesis requires glycine. Purines, creatine. All of the above is the correct answer. In non-ketotic hyperglycemia, which finding is characteristic? 3, 2, 1. And the answer is elevated glycine in blood, CSF, and urine. Thank you. So this was all about glycine metabolism. So try to figure it out how much you have scored in this MCQ test. If the score is more than 80%, it is very good. More than 50%, it is better you rewatch the video and retake all these MCQs. If it is less than 5, you need multiple watching of the videos, and multiple retakes are needed. So the next video will be on methionine and homocystinurias. Thank you. Please do like, share, and subscribe.